Hello. Uh, Good morning, girls. All of you are fine. Okay, today I will explain T cell maturation, activation, and differentiation. So this comes under lymphocyte activation. Lymphocyte activation. So you are going to study both the T cell maturation, activation, differentiation, and also B cell maturation. So now we'll start with T cell maturation. So the T lymphocytes, how they are matured and activated and differentiated, we'll study today. So they are derived from the bone marrow by hematopoiesis. They are derived from the bone marrow by hematopoiesis. The pro T cell, progenitor T cells are known as pro T cells, migrate to the thymus, and maturation occurs in the thymus. And then after rearrangement of the TCR gene segments and acquire the TCR, that is T cell receptor, and they undergo to selection process, that is positive selection and the negative selection. Positive selection is known as self-MHC restriction and negative selection is known as self-tolerance. And then they acquire surface CD molecules like CD3, CD4 or CD8, and two populations of the T cells are released to the circulation. One is CD4, another one is CD8. So CD4 is known as TH cell, that is helper cell, and CD8 is also known as TC cell or cytotoxic cells. So this is how they enter from the bone marrow hematopoiesis stem cells, the common lymphoid. So migration to the blood, from bone marrow to the blood, and then to the thymus, and finally the maturation to CD8 and CD4. CD8 and CD4. So this is how they are matured. So this is for just for understanding. So as noted, the thymocytes undergo two selection process in the thymus. One is known as positive selection, another one is known as negative selection. So positive selection is also known as MHC restriction, and the negative selection is also known as self-tolerance. So we'll study what it is, positive selection and negative selection. So positive selection of the T cells, which ensures MHC restriction. So MHC restriction, positive selection of the T cells. We'll see what it is. So this is the T cell precursor. T cell precursor. So a rearrangement of the TCR genes takes place and will obtain immature thymocytes with the receptors, and this goes for selection. So positive selection of the cells whose receptor binds to the MHC molecules. So the immature thymocytes binds in the thymus region to the epithelial cells of the thymus, to the epithelial cells of the thymus, to the class 1 or class 2 MHC molecules, so which binds. So the binding of these immature thymocytes to the epithelial cells, that provides a signal. uh, that signal uh, protects the thymocytes from apoptosis. Okay, so those cells which are not binding, so not binding to the MHC molecules, not interact with the 1 minute. Mm, 1 minute. Okay. So the positive selection of the cells whose receptor binds to the MHC molecules. So to this, so the binds to the receptor molecules which survive, and the cells which do not interact with the MHC molecules undergo death by apoptosis. Death by apoptosis. So the cells which interact with MHC molecules survive, and cells which do not interact with the MHC molecules undergo apoptosis. So next, so this is what is given. So what is positive selection? The cells which interact with the interaction allow the immature thymocytes to receive a protective signal that prevents them from undergoing the cell death. Cells whose receptors are not able to bind the MHC molecules would not interact with the thymic epithelial cells, so they consequently would not receive the protective signal, so that leads to their death by apoptosis. Next, coming to the negative selection. So negative selection ensures self-tolerance. So positive selection leads to MHC restriction, and negative selection leads to self-tolerance. So we'll see what it is. So once the positive selected molecules are obtained, so this one, positively selected molecules which have both the population of MHC restricted thymocytes, that is positively selected molecules that survive positive selection, comprises some cells with low affinity receptors for the self antigen presented by the self MHC molecules, and the other cells with high affinity receptors. That means positively selected cells will have both low affinity to the self MHC molecules and also have the high affinity to the MHC molecules. Both have low affinity and okay. So negative selection. So both have high affinity and the low affinity, low high affinity receptors and have the low affinity receptors. So the thymocytes undergo negative selection by an interaction with the thymic stromal cells. That means the latter means with those which have high affinity to the self-MHC molecules, they undergo negative selection by an interaction with the thymic stromal cells. So during negative selection, dendritic cells and the macrophages bearing the class 1 and class 2 MHC molecules, they interact with the thymocytes bearing the high affinity receptors for self-antigen plus the self-MHC molecules or for the self-MHC molecules alone. So this leads to the this negative selection leads to the death by apoptosis. So tolerance to self-antigens encountered in the thymus is thereby achieved by eliminating the T cells that are reactive to these um, that are reactive to these cells. So this is the one. So these are the positively selected molecules, thymocytes, which have both high affinity and low affinity. So low affinity molecules will mature. Right? Low affinity molecules will mature into CD4 and CD8 lymphocytes, and those cells which have high affinity receptors for the self-MHC molecules or for the self-MHC plus the self-antigen, so they interact with the macrophages and the dendritic cells and undergo death by apoptosis. So these are the low affinity molecules which mature, and the high affinity molecules for the self-antigen or self-MHC molecules. So these undergo death by apoptosis. Okay, so this is known as negative selection that gives self-tolerance. Self-tolerance. So this is about the negative selection and positive selection of the thymocytes. Next, T cell activation. THL, that is T helper cell activation. So T cells, which recognizes the specific antigens presented with the MHC molecules on the surface of APCs by TCR, that is T cell receptor. Each T cell has 10 to the power of 5 TCRs on its surface, all of which recognize a one antigen molecule or one epitope. So 10 to the power of 10 clones of T cells will recognize 10 to the power of 10 antigen, but only one antigen. So antigen recognition by TCR provides a stimulatory signal 1 and signal 2. So stimulatory signal 1 to the T cell. So binding of B7 molecule on APC, this is the B7 molecule on the APC with CD28 molecule on the T8. So this is the CD28 receptor and this is the B7 molecules. These are the receptor. So interacting with 28 with the B7 molecule on the T cell, which provides the co-stimulatory signal that is known as the signal 2 to the T cell. So when T cells receive both the stimulatory signal 1 and co-stimulatory signal that is 2, they are activated and leads to clonal activation. Okay, so which gets stimulatory signal plus co-stimulatory signal that leads to activation. Okay, so stimulatory signal without the co-stimulatory signal that leads to clonal energy. Okay, so if the T cells receive only the stimulatory signal without the co-stimulatory signal, they are permanently inactivated. So the T cells are not activated, they are inactivated, and that is known as clonal energy, not energy, it is energy. Next, T cell activation by superantigens. Superantigens are viral or bacterial proteins that bind simultaneously to the V beta domain of the TCR. So you have already studied the structure of TCR. So this is the beta domain of the TCR and to the alpha chain. This is the alpha chain of the MHC molecule. Okay. So this superantigens bind simultaneously to the V beta and alpha of the MHC molecules. So this is the superantigen. So they bind outside of the TCR antigen binding cleft. This is the TCR binding cleft, but this superantigen binds outside of the antigen cleft. Any T cell expressing a particular V beta sequence will be activated by the corresponding superantigen. So hence, the activation of TH cells by superantigens is polyclonal and can affect a huge number of TH cells. So it results in the overproduction of cytokines from the TH cells and from the macrophages. Huge amount of cytokines produce the toxic systemic effects, example shock, vomiting, diarrhea, organ failure, etc., rather than providing immunity. So the superantigens are usually soluble proteins. They're usually soluble proteins, okay, and they are secreted by the bacteria or viruses. So these are the some of the superantigens given and what diseases they cause. So this is about the T cell activation. Next, differentiation. So activated T cells generate the effector cells and the memory cells, E cells and the M cells. So this is how they are differentiated. So activated T cells start proliferating. So once the signal 1 and signal 2 is given, so they proliferate several divisions and gives M for memory cells and E for effector cells. So activated T cells start to proliferate, synthesize and secrete IL-2, that is the cytokine interleukin 2, and express the IL-2 receptors on the cell surface. IL-2 receptors on the cell surface. These are the receptors. These are IL-2 produced. So after several divisions, they differentiate to the effector and memory T cell populations. Memory T cells have the long lifespan, 20 to 30 years, and provide immunity if the person is re-exposed to the same antigen. So memory T cells provides immunity and they have longer lifespan, but the effector T cells are short-lived cells and carry out the specialized function. CD8 effector T cells that induce the apoptosis of the virus infected and tumor cells, and CD4 effector T cells that secrete cytokines that cause macrophage activation to kill the intracellular pathogens and also to help the T cell and the B cell activation. So some of the antigen presenting cells which are the characteristic co-stimulatory properties, the dendritic cells, macrophages and B lymphocytes. So the stimulatory effect produced is given in this. So this is about the T cell maturation, activation and differentiation. Okay, so you go through once. If any doubts, you please communicate with me. Thank you.